I'm representing a team here at Argonne National Laboratory that is working on miniaturizing adaptive laboratory evolution approaches. Next slide, please. So in order to, you know, uncover improved isolates or engineer improved isolates with better performance, and as Charles mentioned, improved tolerances for various things, and we'll talk more about that in a little bit. We're going to need, new approaches. And this is an approach that we've developed at Argonne National Laboratory based upon microfluidics. And so here we're using a droplet-based approach to do adaptive laboratory evolution and screen directly in drops that are as small as, say, 70 picoliters. And I want to walk through this technology with you in a case where we've actually used it to select isolates of Pseudomonas, making muconic acid, as you've heard a lot about today, where they have extremely high feedstock loadings and can they now tolerate higher loadings so you can get better titer rates and yields out of your strains in the future. Next slide. So how does our approach work? So it sounds sort of crazy to miniaturize this, but it's just a further development of, say, going from flask down to multi-well plates. And here we're now using aqueous droplets in fluorinated oils. And we're able now to take an initial seed of a parent strain of cells and distribute those individually within these small little droplets. And we get cell growth at rates and to densities that are very similar to that you would see at larger scale. If you're looking to evolve your strain into, higher and higher levels of certain components of the biomanufacturing process, whether it be the feedstock itself, or maybe the product or intermediates, or some, toxic chemical that's in either of those sort of solutions, then you can do this with several rounds and get higher and higher tolerances with additional mutations being isolated and giving the cells advantage in these environments. So we set up these cells. An example of that is shown on the right, upper right for Putida, where we actually have their representation of nearly 3 million test tubes in that small little microfluidic tube at the top. That's the yellow oily component on top of the aqueous layer. And you can see on the bottom right how these cells grow and proliferate. And then on the next slide, you'll see how we now look to evaluate them. So if we're employing this microfluidics approach for droplet-based evolution experiments, then we set up these growth experiments and the droplets, and we do multiple rounds. So we get further and further evolved strains that have larger and larger tolerances to what you're interested in improving the process for. And then we can actually do an initial round of screen from those droplets themselves. So we can actually get a feeling for the cell turbidity and say if they're producing something that you're interested in, how do these evolved cells actually compare in their production of the biomolecule interests. And so you further then characterize your strains, and we've spent a lot of time recently then ensuring that what we're seeing at small scale have any relevance to those at volumes that are, important for initial evaluations in bioreactors heading towards pilot scale evaluations and larger. So if what we're doing at small scale has no relevance, then stop. But we'll show you here today how we can get things that are very interesting at small scale that do work out in scale and become of interest. Now we can do it very efficiently with large amounts of variation and do it in a relatively short amount of time. This is all enabled by the automation that's kind of depicted here in this flow diagram, where all of our evaluations for performance can be automated and done relatively quickly. Next slide, please. So you see, I'm just going to talk about one strain of Putida that's very well advanced with its evolution and engineering before. but that was for normal levels of sugars. We're now going to evolve it on a complex hydrolysate sugar stream at 90 millimolar glucose equivalent to see if we can get these cells to grow better in that type of environment. This was a 12-week campaign that went through about 6 rounds of evolution. And you can see how our miniaturization automation actually drove our evaluation of these strains in the next slide. So here you can see when we take our small cells and get an initial indication of how well these strains are performing, you can test the relative growth and relative performance production of the molecule and plot them out, and then take a subset of them and get more information about them. And in this case, we're actually looking to see if the ones that were on the largest end of the scale for the best production, did they scale and preserve sort of their performance enhancements that you see at small scale. And so we now, Angela, next slide, start evaluating them with more traditional approaches. The picoliter evaluations are using a mass spec. There's a lot more variability there in what we're seeing in the growth and the product analytics. But here you can see in more elaborate evaluations that now have time course information on the multi-well plate level, and leading to shake flask evaluations coming up in the next couple slides. You can see that we're getting important performance enhancements over the control strain, which is shown in the black and red line on the bottom. So we're getting large improvements in growth and or tolerances in growth compared to that of the parent strain, which is really struggling in this environment. Next slide, please. So with these larger scale evaluations, we have more confidence in our analytics, both for the growth shown in the top 2 figures at 2 different scales and the production of muconic acid shown in the bottom. But we're going to show this in different ways, just rather than showing you these production curves. Next slide, please. You can see a comparison of sort of ranked order of these new evolved isolates and how they compare in Violin plots shown at small scale to those at larger scale, and shake flasks shown in the yellow diamonds. And on the right is a ratio of the performance at those 2 scales, and they're really tight ratios as far as their performance enhancements goes. Next slide shows them in a little bit different way. You can see a lot of them compared to their parent shown in the black. A lot of the isolates are not only showing the production enhancements that we selected for. Again, we selected that top right corner initially from our picoliter evaluations. You could look at many different regions of that sort of plot in the future, but they all had similar performance enhancements and a similar performance ranking as we now are evaluating them at larger scale. Next slide, please. So it's easiest to see a comparison of how they were evolved in these small little droplets shown in gray compared to evaluations in multi-well plates shown in green, and then at the 1-layer shake flask level. And we've done some of these now in bioreactors over the summer. You can see how the performance enhancements are preserved. And so what we're seeing at small scale is relevant at larger scales. And so we're really excited about this technology and the use of it in the future. Next couple of slides go over possibilities of using it more in the future, but one last slide on just how our analytics are very important to make sure you have high quality analytics to go along with this, because your confidence in learning about their enhancements are greatly improved if you take a couple extra steps and evaluate them additionally, besides our first picoliter evaluations before you do scaling experiments. But all you see here is that the error gets tighter as your confidence improves. Next slide, please. As with all other adaptive laboratory evolution experiments, we have confirmed from sequencing evaluations that we see a lot of different types of changes. And I don't want to dwell on this slide other than say that, you know, we're seeing large duplication, SNPs, complicated, insertions and deletions or combinations of all these above, as you would expect from adaptive laboratory evolution. And this time was set up for high feedstock loading, but the next slide sort of just reminds everyone that it would just as well be used, this approach, to improve, tolerances to your product, to intermediates, other things that are coming along with the mix and the process. Maybe something you choose just to keep your costs down is not going to be as amenable to tolerance from the organism. Lastly, this has been all patented over the summer and shored up that way. So we're interested in talking more and more to industry about partnerships that could use this approach to evaluate your needs, whatever they may be at the time, from performance to tolerances. So thank you.